Hello everyone, I am Felipe. I'll do this first part of our work. We are group 5. Francis will do the middle part and Aaron will do the last part. The first plant that I'll talk about is Echinodorus grandiflorus, and it is commonly known as leather hat, or in Portuguese, chapéu de couro. It's an aquatic plant from Alismataceae family. It's native from Brazil and found naturally in Paraguay, Uruguay, Argentina, Venezuela, and Florida. It's a perennial rhizomatous plant that thrives in wetlands and it can reach up to 150 centimeters tall. It's commonly known for popular properties for their diuretic, anti-inflammatory, antiseptic, and analgesic properties, as well as phytochemical and pharmacological research. You can make tea with the dry leaves of this plant and even soda. So, the challenges to select these plants are that there's almost no genetic resources. The only resource we could find are with taxonomy, but most of them are morphological. And leather hats are restricted from aquatic habitats, and they might get distance one of them. Climate change can be a challenge as well because it might affect growth conditions, because pollution might affect as well, not just soil pollution but water pollution. And I said uh, earlier about the soda. This is the soda that was I mentioned. It's called Mineirinho, and one of the ingredients are leatherhead. So our proposed research projects might get to these compounds, these medical compounds. So study them are the priority. Um, that because they are already used in folk or traditional medicine. So, knowing the benefits of these compounds, of each of these compounds, sequencing these plants might be the way, because knowing how these plants, how genes can relate to these compounds, can better understand and then search for other plants with the same genes on database to know whether this is an exclusive compound of this plant, or we can find it in other commonly known plants, not that plant that not that much people know about. And then if there's just one plant that might produce some of these compounds, we can produce it in other organisms in a heterologously way, or in other plants, or even in E. coli. So the research challenges that we identify are that there's almost no genetic resources, as I said. Climate change might be a problem too. Ideal growth conditions might be a problem because people don't know that much about these plants, specifically these plants. We might know other aquatic plants like rice, but not this one. Um, and restricted distant environments might be challenging because these restricted environments, aquatic environments, might be distant from each other, so plants might get genetic variability.
So the second plant we're going to talk about is Vaccinium floribundum, that is also called Mortiño here in Ecuador. Okay, so I'm going to talk about a little bit about this plant. So this Mortiño, it's a plant from the family Ericaceae, and it is like a slender shrub that grows basically in the region of the Andes. So it is found in Bolivia, Colombia, Ecuador, Peru, and Venezuela. As I mentioned, it is a shrub, so it can reach only to 2 to 3 meters. And a really important aspect is that it grows at high elevation, so basically between 1800 and 3800 meters above the sea level. So this plant is basically used as food. So it produces like these little berries, these dark like blackberries that can be eaten raw. And it is also a really important plant because it is involved in some of the traditions here in Ecuador. So, for example, it is used for the preparation of the colada morada, that is like a drink that we use to celebrate the Day of the Dead here in Ecuador. We also identify some of the challenges that this plant faces. So, the first one and the main one is the climate change and the anthropogenic pressures. So, basically, this is generating like a lot of damage in their habitat and a lot of fragmentation in their habitat, which is greatly reducing the populations of this plant. This plant is also really susceptible to diseases and to the competition generated by invasive species. We also found that this plant has very few genomic resources, and also that the cultivation of this plant is really difficult. So the harvesting relies mainly on wild plants, and this is also affecting the balance and the ecological role of these plants in the whole ecosystem. As I mentioned before, this is a plant that is really difficult to cultivate. So its harvesting mainly relies on wild plants, and this is a plant that hasn't not been domesticated yet. So our research project is basically focused on identifying the best genotypes that can resist environmental conditions in order to generate like better harvesting or better agricultural practices for this plant. So in this case, we're focusing mainly on drought-resistant genotypes, but the idea is that we can replicate later this methodology in order to focus on different environmental conditions. So the first step will be to collect different individuals from different locations because this plant grows in a lot of regions. In order to get a wide diversity of these plants, we can collect a different individuals from each of the regions. And as I mentioned before, this plant has few genomic resources. So the first step will be to create a reference genome assembly for one of the plants, whatever we collect in the earlier step. So after we have these reference genomes from the rest of the methodology, we can make this RADSEC in order to identify SNPs in all of the plants that we collected earlier in order to like later in the study correlate these SNPs with the plants that have the highest yield in while being stressed in the draft. So the next step will be to stress these plants and select the ones that has the highest yield under these stress conditions. So we can make in this step a QTL analysis in order to identify which are the plants that are more resistant to drafts, and then we can propagate these plants. A complementary analysis that we can make is basically this untargeted LC and MS in order to identify which are the metabolites that are helping these plants to resist better the drought and basically correlate these metabolites to the highest yields. And the idea would be to make this chromatography with all of the plants the worst and the highest yields in order to identify which metabolites are helping the plants best to resist drought. Finally, we also identify some of the challenges that our research will face. So basically, and the main one is the grow conditions. As I mentioned, this plant is really difficult to cultivate because it requires like specific grow conditions, mainly high altitude, soil conditions, and things like that. So it will be a challenge that we need to investigate more in order to generate a better methodology. Then, as I mentioned, it also has few genomic information, so we need to make a lot of steps. First, getting the reference genome and getting a lot of genetic information. So this can also be make our study a lot more expensive. But I think that it is worth it because, as I mentioned, this plant is highly consumed here in Ecuador. So even though the study can be a little expensive, I think that generating and making a better protocol in order to cultivate this plant will be really better and generate a lot of income in the future.
And our third plant, which falls into the ecologically important category, is Thalassia testudinum, or turtle grass. And here you can see a green sea turtle eating both Thalassia and Syringodium seagrasses. Thalassia has a pan-Caribbean distribution. It's in the family Hydrocerataceae, and it's a keystone species, which means that it supports whole food webs in the shallow ocean. This is because it is a nursery habitat for fish, which will go on to live in coral reefs, which are in deeper water. They will also feed manatees and turtles, which are pretty endangered large fauna. And they also support a lot of other fauna, which are food for fish and other predators, like sea urchins, for example. One of the major challenges to coastal habitat in general is eutrophication, which means that there is an excess of nutrients in the water column. This is usually because of people. This is because of sewage or garbage or other forms of pollution, especially agricultural runoff, where you'll have an excess of phosphorus and nitrogen. And this excess nutrient uh, quantity, which is eutrophication, will cause a bloom of algae, which could crowd out seagrasses and essentially smother them. And so here's a picture, for example, of what an unhealthy seagrass bed looks like. You have a few individual plants that are alive, but otherwise, a lot of the other plants have died and have been taken over by bacterial mats. And so the challenges that I'll select for a potential study would be how we can use Thalassia genetics and perhaps its microbiome to deal with these issues of eutrophication and perhaps pathogen blooms, which lead to massive die-offs of seagrass, like happened in the 1980s. Although eutrophication is a major issue in seagrasses, it isn't the excess of nitrogen, phosphorus, and other nutrients which directly harms seagrasses. Of course, like any other plant, more nitrogen and phosphorus isn't always a bad thing. What the major issue is the overgrowth of other organisms, their subsequent death when they become overcrowded, and then resulting hypoxia and also sulfide increases, which can be toxic to seagrasses. So, in order to see which genes might best allow seagrasses to survive hypoxia and sulfide intrusion, which would result from eutrophication, we could do aquarium trials where we would either limit the amount of oxygen in the aquarium or increase the amount of sulfide, and then see which genes seem to correlate with better performance under these conditions. At the same time, I think that the microbiome is a very significant portion of the plant's adaptation to its environment. And there are several studies that are use metagenomics to understand what bacteria are living on seagrass leaves and roots, and they seem to suggest that they have a significant impact on how seagrasses deal with excess nutrient or with toxic chemicals in the environment. So it would make sense to perhaps develop some kind of isolate library, so that way we could test which isolates might allow seagrass to better deal with these stresses, and if possible, what isolates could be applied to the field during habitat restoration efforts to allow faster and more efficient establishment of seagrasses. The greatest challenge of working with Thalassia is its growth conditions. It is a marine plant, so it would require the creation of controlled environments in aquaria, and that in of itself is pretty tedious. It would require near constant monitoring of evaporation and salt content to make sure that there isn't any unequal change in the environment of each experimental replicate. At the same time, if we were to identify genes that seem to help seagrasses deal with eutrophication, or the effects of eutrophication, there is really no method for genetic manipulation of seagrasses at the moment, so there wouldn't yet be a way for us to use that information to perhaps overexpress a gene or underexpress a gene, because genetic manipulation of Thalassia has not yet been developed. Um, at the same time, if we were to look at the microbiome, um, there are many microbiome isolates that would probably be anoxic or microoxic, and thus it would be very difficult to culture them and create a library with those isolates outside of it growing in an aquarium with a plant. So all those things would need to be developed in order to see how we can identify what isolates would benefit the plant under these conditions, and also what we could do with the RNA-seq data once we get it.